Hello, I'm Stephen, and today I'll be presenting modeling protein-stable isotope incorporation in complex microbial communities. Protein-stable isotope probing experiments allow us to connect nutrients to consumers in complex microbial communities. The way this works is we incubate a community in a nutrient that has been synthesized with a stable isotope label like carbon-13 or nitrogen-15. The organisms that eat that nutrient will then incorporate that label into their proteins. We extract the proteins and digest these into peptides and measure them with LC mass spec. Mass spec has two basic kinds of scans. MS1 scans measure all of the masses of all of the analytes that are coming in to the instrument at a specific time, and we can see the pattern of isotope incorporation in those masses. MS2 scans isolate a single mass, fragment that, and the fragmentation pattern allows us to identify what analyte was generating that mass in the first place. There are some existing software tools for analyzing these sorts of data. Cypros is a peptide search engine that allows you to match peptides to MS2 spectra. Unlike other peptide search engines, it's capable of accounting for peptides that have heavy labels incorporated in them. MetaProSIP fits isotope incorporation models to MS1 isotope patterns and uses identifications taken from unlabeled control samples. We have tried both of these tools on an in-house data set that we're working on, and neither of them really worked. This has motivated us to write a new tool, Isopacket Modeler, which allows us to analyze protein SIP data. So what kind of data are we actually looking at here? So we're looking at the MS1 isotope patterns, and a simple model would be every organism that is making a particular peptide is taking up the nutrient at the same rate, and that would give you a binomial distribution of isotope incorporation. We don't see that. We see this sort of more complex pattern where you have a subpopulation of metabolically inactive organisms that are not taking up any label, and another subpopulation of metabolically active organisms that are taking up label, but there's cellular heterogeneity in the label uptake rate, which results in a broadening of these distributions. And we can look at another public data set and see similar patterns where there's a background distribution and a metabolically active subpopulation. We then benchmarked our tool against these other tools. So this is looking at the two data sets that we just discussed. Cypros is by far the least sensitive. It identifies the fewest peptides. Isopacket Modeler is intermediate here, and MetaProSIP identifies the most peptides. We have another public data set that, or we can use another public data set that has E. coli grown to specific label percentages. So we know what the true answer is. We can then use that to estimate accuracy. Cypros, although being the least sensitive, is the most accurate. Isopacket Modeler is again intermediate. And there's quite a number of these where MetaProSIP will regularly fail to properly model these data. There's another public data set that we found that has labeled E. coli spiked into a background of unlabeled bacterial synthetic community. And this allows us to estimate the false discovery rate. Isopacket Modeler, which includes a false discovery rate control step, has by far the lowest false discovery rate of these three tools. So how does Isopacket Modeler work internally? We start with the raw data and the peptide spectrum matches that come from any peptide search engine. And then there's the optional false discovery rate control step, where we fit a convolutional neural network classifier to classify isotope patterns into labeled versus unlabeled. This works by having unlabeled control samples that give us the distribution of what unlabeled isotope patterns look like, and then anything that doesn't fit that distribution is called labeled, which is a positive unlabeled learning strategy. Though the labeled isotope patterns are then fit by between 1 and 4 optional models. We have a binomial model, which states that everything that's producing this peptide is picking up label at exactly the same rate. We have a beta binomial model, which allows for cellular heterogeneity in label uptake rate. And then both of those have an optional unlabeled background, which says that there's a subpopulation that is either not taking up the label or is metabolically inactive. So what can we do with this? So this is going back to the motivating data set, which is a time series of a microbial community. And we can look at we're fitting the beta binomial background mixture model to these data. And we can look at the relative abundance of organisms that are in fact picking up label over time. And this allows us to estimate the growth rate of those organisms. And because we're working with peptides, we know what organism had to have made those peptides, so we can get these growth rates in a taxonomically resolved manner in one pot in an omics fashion. And so what we're finding is that these are very slowly growing organisms. They've got a biomass doubling time on the order of about 250 days. So with that, I would like to thank my collaborators and funders, and thank you for your time.